Hello everyone. EFRUZ cancer carcinogenesis theory and molecular mechanisms. Volume 2 Oxidances, antioxidances and carcinogenesis. EFRUZ ASİLOÐLU, independent researcher. Ascorbic acid, L-ascorbic acid vitamin C is an important vitamin in the human diet and is abundant in plant tissue. Green leaves have the same amount of ascorbat as chlorophyll. Because of its nutritional importance, the distribution of ascorbat has been extensively quantified in plants. However, relatively little consideration has been given to its function in the plant. Ascorbat has been shown to have an essential role in the several physiological processes in plants, including growth, differentiation, and metabolism. Ascorbat functions as a reductant for many free radicals, thereby minimizing the damage caused by oxidative stress, but ascorbat may have other functions which remain undefined. Apparently, synthesis of ascorbat occurs in the cytosol because a specific ascorbat translocator has been identified on the chloroplast envelope. L-ascorbic acid is synthesized from hexose sugar in higher plants, but controversy remains concerning some steps in its synthesis. Although two distinct pathways are possible, higher plants primarily convert D-glucose to ascorbat by a direct conversion that maintains the carbon chain in the same sequence. The pathway involves the oxidation of carbon 1 of D-glucose L-sorbose 1 L-ascorbic acid. Ascorbat can directly scavenge oxygen-free radicals with and without enzyme catalyst, can indirectly scavenge them by recycling tocopherol to the reduced form by reacting with activated oxygen more readily than any other aqueous component. Ascorbat protects critical macromolecules from oxidative damage. The reaction with the hydroxyl radical is limited only by diffusion. Ascorbic acid is synthesized from D-glucose as an antioxidant. Ascorbat will react with superoxide, hydrogen peroxide, or the tocopheroxyl radical to form monodihydroascorbic acid and or the hydroascorbic acid. The reduced form are recycled back to ascorbic acid by monodihydroascorbat reductase and dihydroascorbat reductase using reducing equivalent from NADPH or NADPH or glutathione respectively. Dihydroascorbat may decompose into tartrate and oxalate. The reaction with superoxide may serve a physiological similar role to superoxide dismutase. The reaction with hydrogen peroxide is catalyzed by ascorbat peroxidase. The indirect role of ascorbat as an antioxidant is to regenerate membrane-bound antioxidants, such as tocopherol that scavenge peroxyl radicals and singled oxygen respectively. Tocopheroxyl radical plus ascorbat, tocopherol plus monodehydroascorbat. The above reactions indicate that there are two different products of ascorbat oxidation, monodehydroascorbat and dehydroascorbat, that represent 1 and 2 electron transfers respectively. Monodehydroascorbat can either spontaneously dismutate or is reduced to ascorbat by NADPH monodehydroascorbat reductase. 2 monodehydroascorbat plus dehydroascorbat, monodehydroascorbat plus NADPH ascorbat plus NADP. The dehydroascorbat is unstable at pH greater than 6, decomposing into tartrate and oxalate. To prevent this, dehydroascorbat is rapidly reduced to ascorbat by dehydroascorbat reductase using reducing equivalence. From glutathione, 2 glutathione plus dehydroascorbat, glutathione reducing form, plus ascorbat. Ascorbat has been found in the chloroplast, cytosol, vacuole, and extracellular compartments of the cell of the ascorbat in the mesophyll lip cell is in the chloroplast. The chloroplast contains all the enzymes to regenerate reduced ascorbat from its oxidized products. Foyer and Halliwell proposed that hydrogen peroxide was dissipated in the chloroplast by the coupling of ascorbat and glutathione redox cycling. Illuminated chloroplasts produce superoxide and hydrogen peroxide from site on the thylakoids, most commonly photosynthesis 1. Superoxide is converted into hydrogen peroxide by either spontaneous dismutation or by the superoxide dismutase enzyme. Hydrogen peroxide is scavenged by ascorbat and the enzyme ascorbat peroxidase. The monodehydroascorbat has 2 routes of regeneration, one via monodehydroascorbat reductase and glutathione. The terminal electron donor is NADPH. This pathway serves 2 functions. One is the detoxification of hydrogen peroxide that might otherwise participate in Fenton reactions, and the second in the oxidation of NADPH. The latter function is an apparently energy-consuming wasteful process, analogs to photorespiration. It might at first seem more logical that the chloroplast contain catalase because it would allow the dissipation of hydrogen peroxide without wasting NADPH. However, it should be realized that conditions favoring electron transfer from photosynthesis 1 to oxygen generally cause a high redox potential, etc. High NADPH divide NADPH ratio by reducing this redox potential through the Halliwell-Asada pathway. The tendency of photosynthesis 1 to reduce oxygen is minimized, although ascorbat metabolism has been studied in most detail in the chloroplast. It is likely that all enzymes for its regeneration also exist in the cytosol of both photosynthetic and non-photosynthetic cells. For example, different isozymes for ascorbat peroxidase have been shown to exist in the cytosol and chloroplast compartments. The cell wall is also an important side of ascorbat metabolism because it contains millimolar concentration of ascorbat. Here, ascorbat may have a role in cell wall biosynthesis. The cell wall does not contain ascorbat peroxidase but contain ascorbat oxidase. This enzyme contains 812 copper molecule per enzyme and catalyzes the reaction since the enzyme to recycle oxidized form of ascorbat are not present in the cell wall. It has been proposed that the plasma lemma may have an ascorbat translocator to shuttle oxidized and reduced form between the cytosol and cell wall. Glutathione. Glutathione is a tripeptide. Glutamine, cysteine, and glycine, whose antioxidant function is facilitated by the sulfhydryl group of cysteine. On oxidation, sulfur forms a thiol radical that reacts with the second oxidized glutathione, forming a disulfide bond. Some legumes contain homoglutathione, that is homologous tripeptide of glutathione, cysteine, alanine. Glutathione has a redox potential of minus 440 millivolts that enables glutathione to reduce the hydroascorbat to ascorbat or to reduce the disulfide bonds of proteins. Glutathione is found in most tissue cells and subcellular compartments of higher plants. Its levels declines with tissue age and vary among growth environments. For example, glutathione levels are higher in the light than in the dark. At the subcellular level, glutathione concentration is highest in the chloroplast, averaging between 1 and 4 millimolar, but significant quantities also accumulate in the cytosol. Glutathione exists predominantly in the reduced form, with estimates varying from 70% in barely chloroplast to 90% in pea chloroplast. Glutathione can function as an antioxidant in many ways. It can react chemically with singled oxygen, superoxide and hydroxyl radicals, and therefore function directly as a free radical scavenger. Glutathione may stabilize membrane structure by removing acyl peroxidases formed by lipid peroxidation reactions. Glutathione is the reducing agent that recycles ascorbic acid from its oxidized to its reduced form by the enzyme dehydroascorbat reductase. Glutathione can also reduce dehydroascorbat by a non-enzymatic mechanism at greater than 7 pH, and glutathione concentrations greater than 1 millimolar. This may be a significant pathway in chloroplast, whose stromal pH in the light is about 8, and glutathione concentration may be as high as 5 millimolar. There are alternative functions for glutathione in cellular metabolism. Independent of its antioxidant properties, it may have a significant role in the transport of reduced sulfur from leaves to sink tissues, such as the root. Glutathione also participates in the detoxification of xenobiotics as a substrate for the enzymes glutathione S-transferase. The well-documented tolerance of maize to the triazine herbicides is the result of conjugation of the glutathione to the herbicide. Glutathione is also the precursor of the photochelations that act as heavy metal binding peptides in plants. The synthesis and degradation of glutathione occurs continuously through the glutamyl cycle that has been well characterized in animals, and at least portions have been confirmed in plants. The first step is glutathione synthesis, is the combination of glutamate and cysteine to form glutamyl cysteine by the enzyme glutamyl cysteine synthetase. The subsequent step involves the addition of glycine by the enzyme glutathione synthetase in the legumes that accumulate H-glutathione. This second step involves the addition of alanine by the enzyme hemoglutathione synthetase. The degradation of glutathione involves first the cleavage of the bond between glutamate and cysteine by glutamyl transpeptidase and the transfer of the glutamate residues to an acceptor amino acid. Subsequently, the cysteine glycine dipeptide is degraded by dipeptidases and glutathione AA by glutamyl cyclotransferase. Alternatively, plants may contain an additional pathway to that described in animals for degradation of glutathione involving a glutathione-specific carboxypeptidase and glutamyl cyclotransferase. 5-oxoproline plus cysteine. The presence of the alternative degradative pathway in plants is significant because glucosylcysteine is a common intermediate in the synthesis and degradation of glutathione and thus may be a site for regulating glutathione content in plant cells. The final step in the degradation is the conversion of 5-oxoproline to glutamate by 5-oxoprolinase. 5-oxoproline glutathione, the enzyme that catalyzes the synthesis and degradation of glutathione, have been characterized in both the chloroplastic and cytosolic compartments. The numerous inhibitors of these enzymes have been used in animal systems to characterize the function of glutathione in cellular metabolism, but their use in plant systems has not been extensive. Similarly, although a number of environmental stresses have been shown to cause glutathione accumulation, the mechanism involved has not been defined. The reduction of glutathione, reduced form, to glutathione is catalyzed by the enzyme glutathione reductase, which has been purified from a number of plant tissues. Animals have only one form of glutathione reductase. Plants have multiple forms of the enzyme. Glutathione reductase is associated mainly with the chloroplast, but significant activity is also found in the cytosol and a lesser amount in the mitochondria. The different isozymes quite probably represent the products of different genes with slightly or perhaps significantly different sequences and with differing regulation and responses to environmental signals. Animals contain a selenium-containing enzyme, glutathione peroxidase. This enzyme was thought to be absent from higher plants, but recently there have been reports of glutathione peroxidase in cultured cells. The plant cDNA showing complementary DNA showing homology to animal glutathione peroxidase has been isolated from Nicotiana sylvestris. Tocopherol. The tocopherol, specifically a tocopherol vitamin E, have been studied extensively, that is membrane stabilizers and multiphase that antioxidant that scavenge oxygen radicals, lipid peroxyradicals, and single oxygen. This role is related to its hydrophobic nature, dictates that tocopherol is located exclusively in cell membranes and is oriented with the phetyl chain associated with the fatty acids in the hydrophobic inner regions of the membrane bilayer. The ring oxygen is near the surface of the membrane bilayer but remains exposed to the lipid environment. Tocopherol has been found in all higher plants in both photosynthetic and non-photosynthetic tissues. Although tocopherol is a well-documented component of chloroplast membranes, there are no quantitative estimates of its distribution among other plant cell membranes. Tocopherol is actually a family of antioxidants that includes 4 tocols that have a phetyl chain and analogs, tocotrienols that have a geranyl chain. A tocopherol is generally considered to be the most active form of the tocols. The other forms may be biosynthetic precursors. The relative synthesis of the tocols and tocotrienols may depend on the relative availability of phetyl and geranyl precursors. The tocopherol synthesis occurs in plastids with the aromatic ring formed by the shikimic acid pathway and the phetyl chain synthesized from geranyl pyrophosphate through the terpenoid pathway on the plastid envelope. The latter forms a common link among the synthesizers of chlorophyll, tocopherol, and carotenoids. A tocopherol is well established as a membrane stabilizing agent. Although some of this activity is due to its influence on membrane lipid organization, at least part of this activity is the result of its ability to complex free fatty acids. Free fatty acids act as detergents in membranes, causing disruption of the bilayer membrane aggregation and fusion. The association between the carboxyl group of the fatty acid and the ring of tocopherol reduces this destabilization. The antioxidant properties of the tocopherol are the result of its ability to quench both single oxygen and beta-carotene, and therefore in the thylakoid membrane it may function to break carbon radical chain reactions by trapping peroxyl radicals. ROO plus tocopherol, ROOH carboxyl plus tocopherol, the tocopheroxyl radical is stabilized by the fully substitute benzoquinone ring and therefore does not propagate the radical reaction, making tocopherol an effective radical trap. Because the active oxygen of the alpha tocopherol is located near the surface of the bilayer, and because it readily diffuses laterally in the plane of the bilayer, tocopherol can react with peroxyl radical formed in the bilayer as they diffuse to the aqueous phase. This position also allows the tocopheroxyl radical to be reduced by ascorbat in the aqueous phase to regenerate the tocopherol. Glutathione reduces tocopheroxyl radical in alcohol solutions, but there is only limited evidence for this in biological systems. Structure of tocopherols and tocotrienols commonly found in plants. Carotenoids. Carotenoids are C40 isoprenoids and tetraterpenes that are located in the plastids of both photosynthetic and non-photosynthetic plant tissues. In chloroplast, the carotenoids function act as accessory pigments in light harvesting, but perhaps a more important role is their ability to detoxify various forms of activated oxygen and triplet chlorophyll that are produced as a result of excitation of the photosynthetic complexes by light. There are two classes of carotenoids. The carotenes are hydrocarbons. The xanthophyls are carotene derivatives that contain one or more oxygen atoms. Carotenoids are synthesized from geranyl pyrophosphate from the isoprenoid pathway in plastids and thus have common precursors to chlorophyll and tocopherol. Beta-carotene is formed by the cyclization of lycopene, and the xanthophylls are formed by mixed function oxidases that introduce hydroxyl groups to the carotene molecule. Details of these biosynthetic pathways have been recently reviewed. Certain classes of herbicides function by the inhibition of carotenoid biosynthesis. The carotenoids can exist in a ground state or in one or two excited states after the absorption of light energy. Details of carotenoid photochemistry are given elsewhere. In terms of its antioxidant properties, carotenoids can protect the photosystem in one of four ways: by reacting with lipid peroxidation products, to terminate chain reactions by scavenging single oxygen and dissipating the energy as heat by reacting with triplet or excited chlorophyll molecule to prevent formation of single oxygen or by the dissipation of excess excitation energy through the xanthophyll cycle. Carotenoids may augment the tocopherol is scavenging peroxy radicals beta-carotene plus R-double-O beta-carotene plus R-carboxy beta-carotene plus ROO inactive products. These reactions therefore act as chain terminations, but unlike tocopherol, a mechanism to recycle beta-carotene to the reduced form has not been described. The main protective role of beta-carotene in photosynthetic tissue may be through its direct quenching of triplet chlorophyll, which would prevent the generation of single oxygen and therefore completely avoid oxidative stress. Energy is transferred from chlorophyll to a carotenoid, which subsequently dissipates the energy in a non-radiative form, heat. Thus, the carotenoids act as a competitive inhibitor for the formation of single oxygen, and this is aided considerably by their proximity to chlorophyll in the light harvesting complex. This method of protection is especially critical as light intensity increases above saturating levels. Another carotenoid, zeaxanthin, has been implicated in the dissipation of thermal energy, but the precise mechanism has not been resolved. Zeaxanthin apparently facilitates the conversion of the xanthophylls between two forms, violaxanthin and zeaxanthin. The deepoxidase enzyme catalyzes the deepoxidation of violaxanthin to zeaxanthin in the presence of excess light, and an epoxidase catalyzes the reverse reaction in the darkness or low light. Zeaxanthin therefore accumulates under light intensity that exceed photosynthetic capacity. The deepoxidase has low pH optimum 5.1, whereas the epoxidase has a high pH optimum 7.5. Reduced as ascorbat serves as the electron donor for the epoxidase, whereas NADPH supplies reducing equivalent for the epoxidase structure of two common carotenoids found in plants, beta-carotene and zeaxanthin. The xanthophyll cycle for the cycling of violaxanthin and zeaxanthin. The deepoxidation reaction converting violaxanthin to zeaxanthin is favored under high light and pH 5.1, whereas the epoxidation reaction is favored under low light and pH 7.5. The enzymes for both reactions are in the chloroplast thylakoid lumen, and therefore during periods of photosynthesis the lumen is acidified and zeaxanthin accumulates. The reverse reactions occur in the dark and violaxanthin accumulates. Herbicide tolerance. A number of herbicides that are widely used on many agricultural crops act by the production of oxygen free radicals. A few plants have evolved or have been selected for tolerance. In some plants is related to the plant's ability to scavenge activated oxygen and thereby avoid oxidative stress. Two examples will be discussed in detail. Paraquat. Paraquat and diquat are piperidillium biologen herbicides that are effective as non-selective herbicides when applied to leaves. Symptoms include rapid wilting and desiccation of the leaf, followed by necrosis with 24 hours. Both light and chlorophyll are required for rapid response to the herbicide, which suggested to the early researchers that the herbicide was acting on photosynthesis. In the dark and in etiolated seedlings, the symptoms appear more slowly. At the ultrastructural level, the first injury symptoms are chloroplast swelling, followed by breakdown of tonoplast and the plasma lemma, and then breakdown of the thylakoids and chloroplast envelope. These observations indicated that cellular organelles differ in their sensitivity to the toxic agent, and that the toxic agent is able to diffuse within the cell from its site of production in the chloroplast to the tonoplast and plasma lemma, where it has its action, promotes the visible symptoms of wilting and necrosis. That the toxic agent is some form of activated oxygen was suggested by the first experiments of Mies, who postulated that hydrogen peroxide was produced from the photosynthetic electron transport system. This was based on the observation that moneuron, an inhibitor of the Hill reaction, slowed the phytotoxic symptoms. Calderbank proposed that the hydroxyl radical was produced by these reactions, and subsequently Parrington et al. Suggested the involvement of superoxide paraquat has a redox potential of 446 millivolt, which is critical to its function as a free radical generator. Any reducing agent with sufficient energy can donate an electron to the bipyridylium radical to form the original paraquat 2+, results in the transfer of the electron to oxygen and the formation of superoxide. Subsequent Haber-Weiss and Fenton reactions yield toxic hydroxy radicals. The paraquat ion +2 is again ready for another cycle, and thus the herbicide functions as a catalyst to transfer reducing equivalents to oxygen in the leaf. The major reducing agent with sufficient energy to reduce paraquat is the primary electron acceptor in photosynthesis 1. Ferrodoxin, but other reducing agents may be effective. Consequently, bipyridylium herbicides are also quite toxic to non-photosynthetic organisms, including humans. Resistance to paraquat has appeared in a number of weedy species after prolonged use of the herbicide Fürst and Vaughan. In Lolium perenne, after breeding and selection, Harper and Harvey, and in genetically engineered plants, animal cells, and microbes. In paraquat sensitive plants that have been selected in the field by prolonged exposure paraquat, the mechanisms of resistance is ambiguous. In biotypes of Coniza barasiensis, Chaltiel and Gressel, and in perennial ryegrass Lolium perenne, Harper and Harvey, resistance was correlated with increased activity of superoxide dismutase and other free radical scavenging enzymes. However, in other resistant plants there is reduced translocation of the herbicide to its site of action in the chloroplast, and in other instances correlation with superoxide dismutase activity was not observed. Unfortunately, the situation is just as ambiguous in genetically engineered organisms that have modified expression of superoxide dismutase or glutathione reductase. Gruber et al. Expressed the E. coli manganese superoxide dismutase in the Cyanobacterium anastitis nidulans and observed that the transformants had less diminished. when treated with paraquat. Similarly, Bowler et al. Expressed a manganese superoxide dismutase complementary DNA from Nicotiana plumbigifolia in N. tabacum and showed reduced cellular damage when the transformed plants were treated with paraquat. However, tomato and tobacco plants that were transformed with copper zinc superoxide dismutase complementary DNA from Petunia did not have enhanced paraquat resistance. Superoxide dismutase is unlikely to be the only enzyme involved in providing protection against activated oxygen produced by paraquat. Glutathione peroxidase has been associated with paraquat resistance in a human cell line. Human cell line 60. A transgenic N. tabacum plant that expressed the glutathione reductase gene from E. coli had reduced susceptibility to paraquat compared to the control plant based on visual leaf damage. Malan et al. Observed that in maize inbreds the tolerance of paraquat and also drought was not significantly correlated with the activity of either superoxide dismutase or glutathione reductase individually, but it was correlated with the two enzyme activities collectively. Glutathione reductase reduces production of reduced oxygen by isolated chloroplast in the presence of paraquat, presumably by restraining of the NADP pool. There are also developmental changes in the plant's tolerance to paraquat. For example, in winter, wheat mesophyll protoplast, excised leaves and seedlings have increased tolerance to paraquat after acclimation of the seedlings to centigrade. This was associated with an increased quantity of lipid-soluble antioxidants that would minimize the deleterious chain reactions associated with lipid peroxidation. The above discussion illustrates a generalization which will be a common theme in the chapters to follow. Most of 10-hour experimental research involves simple experiments that have focused on individual components of stresses tolerance, in this case herbicides tolerance. The plant is not so simple and as a general rule will not develop a single method of surviving a potentially catastrophic stresses. If two or more can be developed in parallel, in some instances discussed later, this mechanism of tolerance may be mutually exclusive, but very often they are complementary and serve as backup systems, insurance, or additive protection. Looking at the case in point tolerance of the free radical generating herbicide paraquat, it is obvious that the number of mechanisms may be involved: reduced translocation of the herbicide to its site of action, altered redox potential of photosynthesis 1, primary electron acceptor, so that the paraquat is not an efficient electron acceptor and will therefore not generate superoxide. C. Enhanced scavenging of superoxide produced by photosynthesis 1 and the enhanced protection of membrane components from lipid peroxidation reactions. All the above mechanisms except have been described in tolerant plants. Presumably mechanism is not viable because changes in the redox potential would also change the efficiency of electron transfer between ferrodoxin and NADPH, and this would reduce photosynthetic efficiency and competitiveness or yield. The actual mechanism selected will undoubtedly vary among plants. The redox cycling reaction of paraquat that causes superoxide production. Photosensitizing herbicides. Several herbicide formulations promote the accumulation of metabolic intermediates of chlorophyll, namely tetrapyroles. In the light, photons are absorbed by the tetrapyroles and the energy used to create singlet oxygen that kills the plant. Two classes of photobleaching herbicides, the p-nitrodiphenyl ethers and aminolevulinic acid based modulator, differ in the precise mechanisms of tetrapyrrole accumulation. The p-nitrodiphenyl ethers, exemplified by acifluorphen, are used as selective herbicides in soybeans and require light for herbicidal activity. Early symptoms of injury include wilting, desiccation, and necrosis. Ultrastructural studies have shown that the plasma lemma and tonoplast are the first organelles to be disrupted, not the chloroplast, which explains why enhanced electrolyte leakage is one of the first indicators of injury from a photosensitizing herbicide. Acifluorphen selectively inhibits protoporphyrinogen oxidase protox, which is the penultimate step in heme and magnesium protoporphyrin IX synthesis on the chloroplast envelope. The blockage results in the accumulation of its substrate, protoporphyrinogen, which at the time was thought to oxidize to its product, protoporphyrin 9, by a spontaneous reaction. The loss of enzymatic control of protoporphyrin 9 synthesis was associated with lower Heme levels and increased carbon flow into the pathway. Because Heme is a feedback inhibitor of aminolevulinic acid synthesis, this change in the regulation of the pathway was thought to lead to the 100-fold accumulation of protoporphyrin 9, but a detailed evaluation of the data from these early experiments and acifluorphan action were inconsistent with the herbicide acting solely via tetrapyrrole accumulation, and other mechanisms have been proposed. Subsequently, in vivo localization studies have shown that tetrapyrroles accumulate at sites outside of the chloroplast, such as the plasma lemma, which may explain why photosensitizing damage is not seen initially in the chloroplast. The presumed spontaneous oxidation of the protoporphyrinogen 9 to the protoporphyrin 9 is apparently accomplished by a protoporphyrinogen oxidizing enzyme outside of the chloroplast, probably on the plasma lemma. This enzyme is presumably less sensitive than the chloroplast enzyme to the nitrodiphenyl ether inhibitors, and the presence of this second enzyme would explain many of the inconsistencies of the earlier data. The observed accumulation of protoporphyrin 9 outside of the chloroplast and the observed depletion of the Heme in the chloroplast, the major mechanism of action of the acifluorphan and other diphenyl ether herbicides is now thought to involve a blockage of the chloroplastic pathway, shunting the protoporphyrinogen to an extra chloroplastic pathway that first uncouples the regulation of the tetrapyrrole pathway. On the chloroplast envelope by low Heme levels, and secondly causes the accumulation of a photosensitizing agent. Protoporphyrin 9 in the cellular membranes, the mode of action of the aminolevulinic acid-based herbicides is more clearly established, because unlike other herbicides, they were designed based on a knowledge of chlorophyll synthesis and free radical chemistry. All formulations are based on the concept of supplying aminolevulinic acid to the plant as a precursor of tetrapyrrole synthesis in conjunction with a modulator that qualitatively and quantitatively alters the patterns of tetrapyrrole accumulation by selective stimulating or inhibiting various steps in the biosynthetic pathway. The accumulated tetrapyroles serve as photosynthesizers that produce single oxygen in the light. The herbicide formulations are most effective if the sprayed plants remain in the darkness for a few hours to allow tetrapyrrole accumulation before exposure to the light. After about 20 minutes illumination, the leaves develop isolated bleached areas that expand, followed by wilting, desiccation, and necrosis. Although the photosynthesizing herbicides were assumed to be non-selective, laboratory and field trials have indicated that significant selectivity can be achieved by modifications of the formulation. This is caused by different capacities of tissues and plants to accumulate tetrapyroles and presumably to sequester or translocate the herbicides. Details of the mechanism of the selectivity are given elsewhere. Some plants have apparently achieved increased resistance to acifluorphan by an enhanced scavenging capacity of oxygen radicals. Bean leaves treated with acifluorphan responded by higher level of reduced glutathione and higher activity of glutathione reductase. A tobacco lines that was resistant to paraquat was also co-tolerant of the acifluorphan. This tolerance was associated with the greater inducibility of several components of the oxygen radical scavenging system. Winter wheat, Triticum aestivum, seedling that development tolerance of freezing and of paraquat as they acclimate at 2 centigrade. Also develop increased resistance to acifluorphan. Alfalfa plants that express transgen of the manganese superoxide dismutase complementary DNA from Nicotiana have an increased LD50 for lethal dose 50 for acifluorphan. The proposed site of action of diphenyl ether herbicides is the protoporphyrinogen oxidase on the chloroplast envelope. Inhibition of this enzyme has two effects: reduce HEMA level, which increases carbon flow into the pathway by reducing its feedback. Inhibition of the pathway diverts protoporphyrinogen 9 to the plasma lemma, where the second oxidase converts it to protoporphyrin 9. Aminolevulinic acid based herbicides stimulate the pathway by supplying excess substrate, and modulators promote the accumulation of chlorophyll precursors that act as photosynthesizers. Summary and conclusion. Oxygen radicals are activated. Oxygen has been implicated in diverse environmental stresses in plants and animals, and appears to be common participation in most, if not all, Degenerative conditions in eukaryotic cells, the peroxidation of lipid, the cross-linking and inactivation of proteins, and mutation in DNA are typical consequences of free radicals. But because the reactions occur quickly and often are components of complex chain reactions, we usually can only detect their footprints. Activated forms of oxygen are important in the biosynthesis of complex organic molecules, in the polymerization of cell wall constituent, in the detoxification of xenobiotic chemicals, and in the defense against pathogens. Thus, the plant's dilemma is not how to eliminate the activation of oxygen, but how to control and manage the potential reaction of activated oxygen complex system of scavenging activated oxygen by competing for the energy leaked from the photosystem. Other components are lipid soluble and reside in the membrane bilayer to terminate the lipid peroxidation chain reactions. Still others, ascorbat and glutathione, are aqueous scavengers that detoxify activated oxygen directly or serve to recycle other protective components back to their reduced state. The enzymes that catalyze the synthesis, degradation, and recycling of these antioxidants are essential to viability. Consequently, they are highly conserved among plants and exist in multiple forms in different subcellular compartments and different tissues to allow precise regulation. There are numerous sites of oxygen activation in the plant cell which are highly controlled and tightly coupled to prevent release of intermediate products under stress situations. It is likely that this control or coupling breaks down and the process dysfunctions, leaking activated oxygen. This is probably a common occurrence in plants, especially when we consider that the plant has minimal mobility and control of its environment. These uncoupling events are not detrimental provided that they are short in duration and that the oxygen scavenging system are able to detoxify the various forms of activated oxygen. If the production of activated oxygen exceeds the plant's capacity to detoxify it, deleterious degenerative reactions occur, the typical symptoms being loss of osmotic responsiveness, wilting, and necrosis. At the subcellular level, disintegration of membranes and aggregation of proteins are typical symptoms. Therefore, it is the balance between the production and the scavenging of activated oxygen that is critical to the maintenance of active growth and metabolism of the plant and overall environmental stress tolerance. oxidizing free radical and reactive oxygen species. Indeed, in the biological world, our lives depend upon a delicate balance of oxidation versus reduction, although on this planet, at this time, usually the oxidative process gets the upper hand in our bodies, resulting in accelerated aging for us life forms due to damaged DNA and cells forming tissue, cross-linking of tissues, the accumulation of oxidized end products such as lipofunctions, and finally accumulation of damaged oxidative end products, many of which are substances known as AGE, advanced glycolization end product. Some sources spell the second word as glycation or glycolization. Plasma glutathione peroxidase is one of 4 known selenium-containing human peroxidase. Plasma glutathione peroxidase in E. coli, this has to be done with a mutant where selenocysteine is exchanged for cysteine or serine. This because the translational mechanism for insertion of selenocysteine in prokaryotes are not the same as in eukaryotes. There are some proteins found in different species that have a certain sequence identity with these peroxidases but don't contain the selenocysteine. They are, for instance, found in mouse epididymis, citrus sinensis plant, Nicotina sylvestris plant, and Rugia fungi nematode. Another interesting point of view is the regulation of the sex incorporating factors, an idea to try and make some sort of gel shift study between the stem loop RNA and a lysate occurred, but was already done by Shen et al. In a study, they made an RNA gel shift assay where they found 3 retarded bands and can demonstrate at least 2 proteins binding to the stem loop structure. Plasma nitrate plus nitrat levels and erythrocyte superoxide dismutase activity were higher in the patient group than healthy subject. Patient is leishmaniosis. In contrast, erythrocyte catalase and glutathione peroxidase activity were lower. It was believed that free oxygen radicals were the most important part of the cellular immune response involved in killing the parasite. Recent studies demonstrated that reactive nitrogen radicals represent the main mechanism in the elimination of parasites from the body. Plasma nitrate plus nitrat concentrations and erythrocyte superoxide dismutase activity were found to be higher in cutaneous leishmaniasis patients when compared to healthy individuals, while erythrocyte catalyzed and glutathione peroxidase PX activity were found to be lower in the patient group than in the control group. Mitochondria are the power plants in our cell. Some mitochondria respiratory chain oxygen is partially reduced to form superoxide. Superoxide is radical, a chemical species with an unpaired electron. Radicals usually are very reactive species because electrons like to pair up to form stable two-electron bond. Because of its radical character, superoxide is also called a reactive oxygen species. The production of superoxide by the mitochondrial respiratory chain occurs continuously during normal aerobic metabolism. It has been estimated that 1 to 2% of all the electrons traveling down the mitochondrial respiratory chain never make it to the end, but instead from superoxide. In addition to the mitochondrial respiratory chain, there are other endogenous sources of superoxide production, in the particular when leukocytes, white blood cells, encounter microorganisms or other pathogens invading our bodies, they start to generate large amount of superoxide. Additionally, there are a few external sources of superoxide, especially cigarette smoke, which is packed with free radicals and reactive oxygen species. However, most environmental pollutions other than cigarette smoke and possibly ozone don't contribute significantly to the total load of free radicals and reactive oxygen species to which we are exposed, or to cancer rates either. Under normal metabolic conditions, each cell in our body is exposed to about 10 times 10 molecules of superoxide each day. For a person weighing 150 pounds, this amounts to about 4 pounds of superoxide per year. A substantial amount once forming superoxide is converted to other reactive oxygen species. In the presence of small amounts of iron or copper, hydroxyl radicals may be formed. Hydroxyl radicals are extremely reactive and can cause severe damage to cells and tissue. The recent, though as yet unconfirmed, evidence that excess iron intake may be linked to increased risk of heart disease and cancer may in part be explained by the role iron plays in the conversion of superoxide to hydroxyl radicals, resulting in oxidative damage to arteries and genes. Oxidative damage The reactive oxygen species formed by the mechanism explained above can cause oxidative damage to various biological molecules. For example, hydroxyl radicals can damage cell membranes and lipoproteins, the particles carrying cholesterol and fat in the bloodstream, by a process called lipid peroxidation. Lipid peroxidation occurs by a radical chain reaction. Once started, it spreads rapidly and affects a great number of lipid molecules. Proteins may also be damaged by reactive oxygen species, leading to structural changes and loss of enzyme activity. An enzyme is a protein that catalyzes the rate of a chemical reaction without itself being changed. Oxidative damage to DNA also occurs at a high rate under normal metabolic conditions. It has been estimated that the DNA in each cell of our body suffers 10,000 oxidative hits per day, leading to the formation of more than 20 different oxidative DNA lesions. Many of these lesions are known to cause mutations. There are a number of DNA repair enzymes that can remove these lesions, but their repair success is not perfect. Therefore, oxidative DNA damage and mutations accumulate with age, which may contribute to cancer. Sources of ROS, reactive oxygen species. Mitochondrial respiratory chain activated leukocytes in indoor and outdoor air, cigarette smoke, radon, ozone, ultraviolet light, etc. Oxidative damage to DNA, lipid proteins, causal contribution to aging heart disease, cancer, Alzheimer's disease, inflammatory, immune injury, autoimmune disease, rheumatoid arthritis, lupus, diabetes, AIDS, adult respiratory distress syndrome, etc. While oxidative damage to DNA is implicated in cancer, oxidative damage to lipids in low-density lipoprotein, LDL, the bad cholesterol, plays an important role in atherosclerosis. Atherosclerosis and the resulting complications of heart attacks and stroke are by far the #1 killer in the United States. Interestingly, only polyunsaturated fatty acid can undergo lipid peroxidation, while saturated fats don't. This is quite ironic because polyunsaturated fat is considered beneficial for keeping LDL levels, low density lipoprotein levels, low in the blood, while saturated fat increases these levels. The solution to the dilemma is to replace both saturated and polyunsaturated fat in our diet with monounsaturated fat, which is neither easily oxidized nor exerts adverse effect on the blood level of low-density lipoproteins. The single most abundant monounsaturated dietary fat is oleic acid, present in olive oil. The high consumption of olive oil may explain why people living in Mediterranean countries like Greece and Italy have such low rates of heart disease. In addition to some other factors, for example, the regular consumption of moderate amount of alcohol. Antioxidant protection. Antioxidants protect us because they can scavenge reactive oxygen species before they cause damage to the various biological molecules or prevent oxidative damage from spreading, etc. by interrupting the radical chain reaction of lipid peroxidation. The antioxidant defense systems in the human body are extensive and consist of multiple layers which protect at different sites and against different types of reactive oxygen species. An important part of the antioxidant defense system inside cells are the antioxidant enzymes, for example, superoxide dismutase scavenges superoxide and converts it to less reactive species. It was recently discovered that the gene for superoxide dismutase is defective in patients with amyotrophic lateral sclerosis, better known as Lou Gehrig's disease. The very existence of superoxide dismutase biological system as well as other antioxidant enzymes, etc. catalase glutathione peroxidase, attests to the importance of oxidative damage as a real threat to cellular and organismal survival. Otherwise, organism would not go through the considerably trouble and energy expenditure of evolving and synthesizing these enzymes. In addition to the antioxidants that play important role in antioxidant defense systems, these small molecules antioxidants are particularly important in blood and the fluids present in the extracellular space, where antioxidant enzymes are absent or present only in small quantities. The small molecule antioxidants include lipid-soluble and water-soluble antioxidants. The lipid-soluble antioxidants are localized to cellular membrane and lipoproteins, whereas the water-soluble antioxidants are present in aqueous fluids such as blood and fluids within cells and surrounding them. Alpha-tocopherol, the biologically and chemically most active form of vitamin E, is by far the most abundant lipid-soluble antioxidant in humans. Recent evidence indicates that the intake of about 200 international units of vitamin E per day substantially lowers the risk of heart disease. A very plausible mechanism for this effect of vitamin E is the protection of low-density lipoproteins against oxidation, which, as mentioned above, is a critical step in the development of atherosclerosis. Vitamin E may also be useful in the treatment of heart disease Other lipid-soluble antioxidants are beta-carotene, a vitamin, a precursor, and related substances called carotenoids, such as alpha-carotene, lycopene, the red color in tomatoes, lutein, and zeaxanthin. However, carotenoids are much weaker antioxidants than vitamin E. It was not surprising, therefore, that beta-carotene supplementation failed to affect cancer and heart disease rates in 3 recent, very large and very expensive studies in Finland, Boston, and Seattle. If anything, lung cancer rates were increased in smokers given beta-carotene supplements, a result that awaits a scientific explanation. Vitamin C, the prominent water-soluble antioxidant, has a number of well-defined biological functions, including collagen, catecholamines, and carnitine biosynthesis. Vitamin C also very effectively scavenges a wide array of reactive oxygen species and free radicals. Our studies, Dr. Paulings, have shown that vitamin C forms the first line of antioxidant defense in plasma against many different types of reactive oxygen species and radicals, and surprisingly also protects low-density lipoprotein more effectively against oxidation than vitamin E. There is also strong evidence from epidemiological studies that an adequate intake of vitamin C can lower the risk of cancer, heart disease, and cataract. We are constantly exposed to reactive oxygen species generated from endogenous and some exogenous sources. These reactive oxygen species react with biological molecules such as DNA, proteins, and lipids, causing structural and functional damage. Oxidative damage accumulates in human tissues with age and can casually contribute to a number of degenerative diseases such as heart disease and cancer. Antioxidants, both enzymatic and non-enzymatic, limit oxidative damage to biological molecules by various mechanisms. Dietary antioxidants such as vitamin C and E significantly contribute to antioxidant defense systems in humans and may help protect us from certain age-related degenerative diseases. Prostaglandins Fk-alpha is known to initiate luteal cell apoptosis in the bovine corpus luteum via its specific receptor on the luteal membrane by inducing intracellular calcium mobilization and the activation of protein kinase C. The incubation of bovine luteal cell with hydrogen peroxide and mercaptosuccinate, the specific inhibitor of glutathione peroxidase, resulted in increased chromatin DNA condensation and apoptotic DNA fragmentation. Analysis of the enzymatic activities of glutathione peroxidase and catalase support the RNA data indicating that the hydrogen peroxide produced produced due to the lack of glutathione peroxidase is a potent inducer of luteal cell apoptosis, reactive oxygen species. Reactive oxygen species, including superoxide, hydrogen peroxide, and hydroxyl radicals, have been reported to be generated in rat and bovine corpus luteum during regression. Reactive oxygen species are produced by electron leakage from the mitochondrial P450 system. And cause irreversible damage to cellular DNA. Meanwhile, the activities of antioxidative enzymes such as superoxide, dismutase, catalase, and glutathione peroxidase change drastically during the estrus cycle and pregnancy. Reactive oxygen species induce follicle atresia and luteolysis. These apoptotic events have been found to be suppressed efficiently by estradiol, ascorbic acid, and other antioxidants. It should be noted that prostaglandin Fc-alpha induced reactive oxygen species production in the corpus luteum via the activation of the protein kinase C and intracellular calcium mobilization. To identify the regulators of luteal cell apoptosis, differential display and northern blot analyses were carried out. In contrast to many antioxidative enzymes, the levels of glutathione peroxidase were found to decline in the corpus luteum just before the start of apoptotic events, suggesting that hydrogen peroxide facilitates luteal cell apoptosis in the bovine estrus cycle. It is well known that superoxide dismutase converts superoxide to hydrogen peroxide, thereby protecting cells from the injury's radicals. Recently, several other groups have reported that oxygen radicals are produced during steroidogenesis by the P450 system in mitochondria. GPX levels decrease throughout the estrus cycle, while the levels of other antioxidant enzymes increase, suggesting that glutathione peroxidase is a key mediator in the promotion of the luteal cell apoptosis. The cytosolic glutathione peroxidase could play a major role in protecting luteal cell from cell death. Hydrogen peroxide treated cells, indicating that glutathione peroxidase, not catalase, is the major scavenger of hydrogen peroxide in the bovine luteal cell. Eventually, the oxidative stress caused by hydrogen peroxide should affect both functional and structural luteolysis during the bovine estrus cycle. The importance of FP, corpus luteum specific receptor expression during luteal regression is strongly supported by a study of FP deficient mouse in which functional luteolysis and parturition failed. Several lines of experimental evidence have demonstrated increase in the amount of cyclooxygenase 2 and endothelin 1, as well as an induction of superoxide production of after prostaglandin epic alpha treatment. The result of the present study suggests that hydrogen peroxide accumulates due to decreasing glutathione peroxide that result in structural electrolysis. One messenger RNA increases in regress corpus luteum. The involvement of oxidative stress in the transcription activities of such apoptosis-related genes remains an interesting subject. It will be necessary to examine the association between FP stimulation and the expression of the antioxidative enzymes and to observe the localization of antioxidative enzymes in corpus luteum.